are we gonna pay for that? Well, that's the question.
Hey guys. Hey Cayman Prep. Thanks for being here. We're gonna get started in about 15 minutes. Hey, Cayman International. But we're going to go live in about 5 more minutes. So we're here. Just hold tight for a couple of minutes before we get started. I'm so happy you're here. Hey, Cayman Prep, is it a year 8 class that's with us? Yeah, okay, I was just checking on our ages. Great, thank you. Just a couple more minutes. And then Cayman International, are you year 4? What year are you guys? Oh, you're muted. Oh, I can't hear you though.
Which classes in prep?
Huh?
Which classes in prep? Because my youngest is in year 7.
Year 8. Hey, Cayman International, can we try getting you guys on the mic just to see if it's working? Because I wasn't able to hear you guys. And Cayman Prep, let's try you on the mic too. Let's just practice, make sure that it's working. Okay, but I'm not hearing you. Yeah, I see that your mic is on. Okay, CIS is grade 2. Okay, so if I can't get the audio working, you guys, I'm going to have you'll have to use the chat on the side. Give me a minute to see what's going on with the audio. Hi, everybody. Give me just a couple more minutes. I'm problem solving our audio, and I want to make sure that we can hear you guys. Oh, okay. Okay, so hey, Cayman Prep, can you try saying something on your microphone?
Hi.
Yes, okay, perfect. I figured it out.
Andy, do you want to try?
Okay, CIS, can you guys say something?
Hi.
Perfect. Oh, it's so good to hear your voices. Okay, let's wait one more minute and we're going to get started. Thank you for your patience, everybody. Okay, got it. That was the problem. John Graces are going to be late. Okay, so they had a chat, like I was like, the audio's not working. Yardstream just has like a little, are you having problems? I just clicked it and immediately a guy came up and solved it.
Wow.
That I've never had that on any of our streaming platforms before. Like boom, boom. Huh? It's called Yardstream. Yeah. Oh, I'm gonna mute myself. Alrighty. Hello, everybody. My name's Todd Bohannon. Hello. It's so good to see you guys. I'm a volunteer educator with REEF, the REEF Environmental Education Foundation. And we're here today for our first live stream from Grouper Moon 2023. Right now I'm on Little Cayman. I just arrived here yesterday after flying on a red eye, which means I flew all night from Seattle to Miami. And then from Miami, I flew here, where I flew to Grand and then from Grand to here. It's amazing. As soon as I got here yesterday, I was able to hop onto the boat with a bunch of researchers and we went out and took a look at the fish. And later on in this live stream, I'm going to share with you guys some of the video that I took. But we have a few guests that I'm going to introduce to you guys today, and you'll have opportunities to ask questions. If you guys have questions during the live stream, you can raise your hand, or you can let your teacher know and they can pop it in the chat, and then I can call on you guys. But we'll also have time for questions at the end. But if you have something that comes up, you want to ask a question, don't hesitate. Just raise your hand. That's what we're here for, to engage with you guys. So I'm really excited that you're here. Thank you so much. The first person I want to introduce you guys to is Dr. Croy McCoy.
Good morning, everyone.
Hi, Dr. McCoy. How are you?
I'm doing great. Yourself?
Good. So the first thing that I like to do is kind of give a big picture of what's happening so far here on the aggregation. So when did you with the DOE? So maybe tell us a little bit about yourself, and then we can do a big picture.
Well, I work at the DOE. I've been there almost 28 years, and I head up most of the projects and the marine side of it, which includes Grouper Moon. We took 2 boats from Grand Cayman over to Little Cayman, our big boat, which came over on Friday, and the smaller one on Saturday. These boats are used to do all of our research in the BRAC and also in Little Cayman.
Okay. And can you just tell, how long have you been with DOE?
I started in 1995, actually. My true age is showing up now. I've been there. Been in doing research on our reefs and fish and mangroves, coral reefs for the last almost 28 years. So,
How did you get into that? What was sort of your path?
well, I was born in Brack. I attended Spotway Primary
Woo!
and the high school. And I started out just diving with my family here in Little Cayman. And the interest just went from there, trying to understand, marine biology, if you want to put it in context, and coral reefs and fish behavior and the ecology of fish and stuff. And then I went off to university after that, just spurred off my interests and understanding oceanic systems.
And so you went to university. What degree did you get?
I did a marine biology, marine science degree.
Where did you go?
Florida, University of Tampa.
Okay.
And then I went on to graduate school in North Wales, University of Wales, to my master's degree.
Mhm.
Then I continued that to do my PhD at Bangor University in the UK, in Northern Wales again. And here I am working with the DOE still and doing research and finding ways that we can help Mother Nature, that we all have not only Nassau groupers, but coral reefs and other species of fish and mangroves and seagrasses in for you all and your kids and the future of these islands.
Well, and so what is your, I guess, title? What is your position on this project? And
I'm one of the, from the DOE side, I'm the PI, principal investigator. And that along with reef with Dr. Bryce, or Professor Simmons now, and Dr. Scott Heppel and Dr. Christy Simmons. So 4 of us runs it from different aspects of it.
And so have you guys been out on the aggregation already?
Yeah, we started from Sunday.
Sunday.
Really when we all kicked into gear, getting everything started, and we were in Brack yesterday.
So what does that mean, getting things started? What are you doing?
Well, with anything you start off the first time, you work with all the crooks and wrinkles and stuff. And nothing works perfect in the world. You can plan as much as you want. Boats break, dive equipment break, research equipment break, and you just got to get everything working and well oiled for the rest of the week leading up to the spawning and everything.
So, as you guys know, the Grouper Moon Project is a collaboration between the DOE in the Cayman Islands and REEF, the Reef Environmental Education Foundation. So there's 2 sets of researchers here on the project every year working on this together. What do you guys do to start? Like, what are you out there doing with those fish?
Initially, we started this project using AI with fish faces, just seem as
Okay, so AI fish faces. We're going to slow down for a moment. Let's start with fish faces. What do you mean that you're going out there and are you looking at their faces?
Every individual fish, though we look the same, just like human beings.
Yeah.
Your face looks different from mine. You look different from someone else. So what we do, we take video with GoPros and all as much fishes as we can possibly get within the dive. And
so you're going down, you've got a GoPro camera and you're taking video of the fish's faces.
Yeah, and what we do, we come back and download that and run it through software, facial recognition, using, we'd say AI, which is artificial intelligence, and it recognizes that fish that returns annually. Before that, we used to use it's one way of also getting population estimates of how much fish is there. But not only that, we're interested in they keep returning each year.
So is it kind of like a fish Facebook?
A bit, yeah.
It's like, I mean, because you're creating your category or your
catalog of
cataloging all the fish.
Yeah, who attends there.
And then you're able to see if they're coming back in subsequent years.
Yeah.
So before when that was happening, that they were doing that just with pictures, but now we're doing it with pans. And you said something about AI. What do you mean that AI is helping?
Just artificial intelligence. We have software that we run the video through that recognizes each fish repetitively, so we know that fish was here last year, and it was here again this year.
So you don't have to you don't have to actually go and look at them. The AI will identify them for
But I
you.
identified it first. Yeah.
And what's the benefit of that for the project?
It's a quick and easy way to collect a lot of data really quick and accuracy.
Yeah.
And it also gives us population estimates. We used to use temporary fly tags with tag and recapture before, and we're sort of trying to move away from that, where it's non-invasive. Even though these are pretty much harmless, it's still.
So these are the tags that Croy's talking about. They're called Floyd tags, and they how many put out? Like 100, I think, every year.
We try to put out 100 right away, first thing when we get here, and let the population mix, and then we go and do counts.
It looks like we have a question. Hey, Cayman Prep, did you guys have a question? If you unmute yourselves, you can go ahead and ask. No? Do you want me to try to unmute you? Oh. What's the question? How many Nassau grouper are there? That's a great question. What was the final number that you guys came up with last year?
Well, when the project started, because of fishing pressure, the low Cayman aggregation was just around 1,000 or so. And it took a lot of work and a lot of science and get a management plan in place. But right now we estimate the populations like 7,000 or 8,000 fish. And the brack was actually down to less than just a few hundred. And that has, following the same pattern as low Cayman, though lag way behind over the years. It took us about 10 years before we started seeing the population increase, and low Cayman has tripled, and brack is upwards of several thousand fish easily from a few hundred. We'll have more estimates at the end of this project and exactly what the population status is of brack, but we would imagine it's going to be up in 3,000 or 4,000 fish
on brack.
On brack.
Really?
Yeah.
Now you guys went out there yesterday, is that right?
Yeah.
And what were you happy with what you saw?
Yeah, very happy. It was just around where we have these listening devices.
Oh, hold on, I'm sorry, I'm going to interrupt you because we have a question. Cayman Prep, what's your question? I cannot hear you. That's straight. Oh, I don't know why I can't hear you. Let me try removing you and bringing you back. For some reason, your audio isn't working now, and I don't know why. It was working a minute ago. I'm so sorry. Can you type it into the chat?
There's a question right there.
So while we're figuring out what's going on with that audio, that's so strange. It was just working. We have a question from the Cayman International School, from Miss Amanda's class. Oh, they're running out. They had run out for, okay. So they said, have they spawned yet?
No. The population keeps increasing right up to the full moon and just after. And the fish, we say hydrate. That's when the eggs get real puffy stomachs. And they usually spawn, though we haven't nailed that down to a fine art yet. They usually spawn 2 or 3 nights after the full moon. Full moon was yesterday.
Okay.
So about 2 more days they should spawn, and they'll spawn every night for about 3 nights. But the population of low Cayman is quite high. It's like probably around 7,000 fish or more, and we're hoping to see more spawning, which they will over several more nights, but that's left up to Mother Nature.
All right. There's a question from Cayman Prep. Do you have one famous fish?
There's several famous fish on the site that return annually. We used to have one named Phantom. He had one side white, one side dark.
Really?
And there's this one that the shark took a big chunk out of him, and he's still there and comes and shows up. And
So he's got like a big bite out of the
big
back.
bite out of them. Every fish, if you look closely to them, there's something different about them. That's how they identify even sharks, whales, and everything. So there's loads of famous fish on there that we see annually that, because Nassau groupers, we consider them the puppies of the sea that can be domesticated, like how you have a little pup or a cat or anything like that. So you recognize them every year because of their features that are different from the rest. And there's lots like that. And you almost feel like they recognize you because they come right up to you, at least a Lower Cayman. The population in Brack is a little more skittish or shy, if you want to call it that. So you don't get nowhere near as you do with the population in Lower Cayman, where they almost want to interact with you, which is a special thing about Nassau groupers.
So I also heard when we were on the boat yesterday, one of the researchers saw a fish called hula.
Yeah, she has like a band halfway down midship on the fish, and she's been around probably 12 or 15 years now. That goes back to the beginning when that fish used to show up. So,
really, that long?
Yeah.
Awesome. So we have a couple of other questions. Cayman Prep's asking, how heavy do they get?
They get fairly large. The largest ones we have are in Brack, maybe because they shy away from people, so they don't get caught as quick.
Right.
But the average weight of the Nassau groupers and off the west end of Little Cayman are probably 12, 14 pound. They're a little smaller. The ones in Brack are a little larger, but they get up pretty heavy.
Well, since we were talking about the Brack, I have some video that you guys shot yesterday on the Brack. I'm going to share some of that really quick. So guys, hold on one moment. I'm going to share some video of the aggregation that's on Cayman Brack. Now, I'm told this is stereo video, but I'm not sure what that means.
When we started the project, we used to use two lasers that were measured to get an estimate of distance when you shine it on the fish. And then we would extrapolate to get the full length of the fish. Stereo video is a bit like your two eyes, how you look at something. You can estimate the distance in between that.
I see.
So we have two cameras that are synchronized on a platform that we go up to the fish and take video of it to get the size classes now. So we moved on to a little more complicated system to get better data. That's what we try to do as researchers. We look for better ways to do things to get more accurate data.
I see. So this is the smaller aggregation on the Brack. But what's different about it in these last few years compared to previous years?
I mean, in the Brack?
Yeah.
Well, for one, the population was so low 10 years ago, and we took a long time to see that increase in the population over there. But
and does that correlate like to catch limits and the protections that have been put on to the?
Yeah, the management plan has slot sizes. We don't take the big breeders or big mamas, whatever you want to call them, and we don't take the juveniles. So the slot size is 16 to 24 inches, or not the teenagers, but they at least reach reproductive status and have a chance to continue to contribute to the population. There's bag limits we put onto it also outside of the season, and it's working really well. I mean, the population is still increasing annually.
So I see there's a question here. So this say goodbye to that video because we're going to take off from there and come back to us. Okay, so one of the questions was how many fish have spawned? So maybe let's just talk briefly about what actually happens.
Well, any Nassau group of that have reached reproductive status shows up at the site. We've proven that with all of over the last 18 years looking at the data. So what we got to realize, your whole reproductive population is at this one site. That's why they get fished so heavily, and that's what leads to this demise of population regionally. Originally there was like 50 plus, and over 30 of them have disappeared because of this fact. And they're predictable, which means they gather at one spot, a certain time, and they just get fished out. And that's the whole reproductive effort for that 1 year. They don't do it every month. They do it once a year, sometimes maybe twice maximum. So in that sense, all of the Nassau groupers that are capable of having babies are going to be at the west end of Lower Cayman, also in the BRAC at the east end, and then Grand Cayman at the east end too.
So does that mean that they all spawn at the same time?
Yeah, they do. It's external fertilization where the female releases her eggs and the male releases his gametes, and they mix together. So we call it a dance. So it sort of twists going up a little bit, and then there's a window of 35 to 45 days where it's planktonic. It just floats in the ocean. The eggs hatch after like 24 hours. They're free swimming, and from there they settle out on our reefs. So
awesome. That was a great question, you guys. I want to thank Dr. McCoy for being here and talking to you guys. And I want to introduce you guys to, was there another question you had before we move on? I see a hand. But unfortunately, Cane Prep's mic isn't working. Okay, if you have a question, you can go ahead and put it in the chat. Oh, here we have someone coming up. No? Okay, that's all right. Anyway, thank you so much for being here.
Thank you for having me.
And we will be sharing video later this week of the actual spawning. And so you'll see what Dr. McCoy was talking about with that sort of dance that you were explaining. All right, thank you so much for being here.
Thank you.
All right, next up, we have another scientist who's going to join us, another principal investigator on the project, Dr. Simmons. Well, hi.
Hi, friends. How are you? Good to see you all. Hey,
Yeah.
my friends. How are you all? All right. I love the energy, you all.
So, kind of big picture, what's happened so far on the project? When did you get here? What's going on?
Yeah, so we've been here for a couple of days now. Our team kind of trickles in. We've got a small plane that we have to take from Grand over to Little Cayman. So sometimes it takes quite a bit of effort and many planes to get all of our people and all of our equipment over to this island to start diving. At the same time that we're bringing people over on the plane, the Cayman Islands Department of the Environment is bringing boats over. So we have 2 different Cayman Islands Department of Environment vessels that we're using for our research here. And
So how many people do you think are here this year on the project?
gosh, we've got close to 20 between the Department of Environment staff, scientists, and researchers, and then our component, which is, of course, some academics, scientists, and educators from Reef. So it takes a village, literally. We've got a huge number of people from lots of different places coming in to do the work.
Awesome.
Yeah, so we're now all here, and we've been diving for the last couple of days, and we've been using the two vessels that the Cayman Islands government brought over. One, which is the RV Seakeeper, it's their large research boat. And that's the boat that we put most of our divers on. That helps us to do a large diving platform so we can do research and diving out at the Little Cayman spawning site. At the same time, for the last 2 days, we've been sending a boat over to Cayman BRAC, and we've been fielding a team of divers over there as well to collect data at the exact same time. So the same time we have divers in the water on Little Cayman, we've got divers over in the water in Cayman BRAC.
Awesome. So Dr. McCoy started talking about that you guys are creating a fish Facebook. Can you talk a little bit about how you're doing that? This is the tool that you're using.
This
right?
is the tool.
So what happens?
Yeah. So you probably all have one of these at home, or you've seen one of these. This is just a GoPro, right? Just a small action camera that's in an underwater case, and we've got it on a selfie stick, right? So of course, we're not actually taking selfies of ourselves most of the time. Sometimes we might, but most of the time, what we're trying to do is we're going to capture images of the sides of fish. Right. So like you and like me and like Todd and Croy, we all look very different. And we can tell, you can tell it's me, you can tell it's Todd, you can tell it's Croy just because of the way you look. Well, inside your head is artificial intelligence, except for it's facial intelligence, right? So because you can tell us apart based on our facial features, we can actually do the exact same with grouper. It turns out that the stripes and patterns on the sides of grouper are consistent across time. They keep their same patterns.
So it doesn't change.
It doesn't change. Right. And so Croy mentioned hula, right? That's a great example.
Yeah.
It's an extreme example of a pattern on the sides of fish, but all fish have unique patterns that maintain over days, months, years. And we can use artificial intelligence along with our GoPro to capture that information and keep track of individuals at the aggregation site through time. So we put a whole bunch of divers in the water, filming a whole bunch of fish, and we take all of that footage back to the lab, and we use artificial intelligence to process that footage so we can keep track of individuals. So the artificial intelligence, the computers say, okay, well, you found Bob the fish, and Bob the fish, we've seen him for the last 5 years. He's grown 3 centimeters. We get all of that information by using just GoPros and selfie sticks underwater.
Now, but you're also tagging fish, right?
We are.
Can you talk a little bit about what that is?
Sure.
And why you're doing it?
Yeah. So Dr. Croy, he mentioned that we are constantly trying to evolve the science that we're doing so we can get new and better data. But it's also important that we try and involve the science we do so we can have as little impact on the fish while they're spawning as possible. Because of course, we want to let them do their thing so they can create as many babies as possible so that the Cayman Islands gets as many new fish as possible arriving back to their population. One of the ways that we collect information on the number of fish that are out there is through these tags. This is just 2 of them. We call them jewelry. And we've been, for the last 10 years or so, we've been tagging a very small number of fish with these things, with these little tags, and we implant them in the back of their fish with divers, using a little spear to poke them in the back. And then those tags are not designed to last years. They're designed to last weeks. We don't want them to last years. All we want is for these to be in the sides of fishes so that we can then send divers down and do counts of fish and keep track of how many of those fish have tags. So here's a little math for you. You guys ready? If I tag 100 fish, if I put 100 fish tags out in fish, and then I count 100 fish of that population, because it's well mixed now, and of the 100 fish that I count, 10 of them are tagged. What proportion of the population is tagged? If I have 100 fish and 10 of them have tags, it's probably 10%, right? And if I know that I've tagged 10% of my population and I put out 100 tags, that means that there's 1,000 fish in the population. Right. So this tagging that we do allows us to come up with a mark recapture estimate of population size. Now, I said we're trying to evolve the science, right? So this is minimally impactful to the fish, but it's still impactful.
Sure.
We want to get rid of the jewelry eventually. And so that's why we're moving to the facial recognition. Because if we can do the same thing just with our cameras and artificial intelligence to keep track of individual fish through time, then we don't need to use tags at all. And so we've been rapidly evolving that artificial intelligence over the last 3 years to start to demonstrate that it will replace these tags. So the hope is by next year, we'll never have to use these again.
Really?
Yeah.
So this may be the last year of tagging.
That's the hope.
Wow. That's amazing. So what happens next? Okay, no, let's not go there yet. What's the typical day for you guys going out there on the boat? Are you guys just in the water constantly, or tell me kind of, how does that go?
How does it go? Sure. Yeah. So as a general rule, we're trying to do, as a team, a whole team, we're trying to do 3 dives a day. We'll do an 8 o'clock dive, a 1 o'clock dive, and then a dusk dive, whenever that happens. Now, not every diver goes on every dive, but collectively we try and field 3 teams of divers per day.
That's a lot of diving.
It's a lot of diving, and each dive lasts we're at the bottom down with the fish for about 25 minutes or so. And then we spend another half that amount of time just getting to the fish and then coming back up to the boat safely. Of course, it's fairly deep diving, so we are very safe about how we do our diving. We take safety very seriously, so that means it takes time to get down to the fish and to get back. So we're doing that 3 times a day. But here's the thing, winter in Cayman is not very predictable.
Right.
So some days, because of the weather, because of the sea state, because of the big waves, we may not be able to get out. Or it may be that we can use the Seakeeper, the large research vessel, but maybe not the small research vessel. So we have to adapt to what the weather is doing and what the fish are doing. So it changes every day. So a huge part of our research is constantly getting together and reevaluating and recreating the plan. So it's a constant plan in motion all the time.
It seems like that's a lot of variables for you guys to be managing at one time. Are you doing all of that in your head, or do you guys do you delegate those jobs out? How do you keep that all together?
Yeah, so it's a team effort,
right,
of course, and there are specialists. So we leave it up to the Department of Environment. If they say we can't take the boat out, we don't take the boat out. That's not our, there are experts at boat operations, and there are experts at doing science, and so we've got a collective of people that all work together. It's really amazing actually to see the teamwork.
Yeah.
Like the fact that you've got roughly 20 people in the field that are all contributing to this project and collectively coming up with game plans and evolving those game plans on the fly. It's really amazing that we're able to pull it off. And it's because we all work together very well as a team.
Yeah, it's amazing. And you guys are part of that team just by being here with us and participating in these live streams. For this project to be successful, we really, it takes everybody to take care of and to conserve something as important as the Nassau grouper and the coral reefs that are here on Cayman Islands. Now, I have some video that I took yesterday of the aggregation that I want to share, and maybe you can talk a little bit about what we're seeing.
Absolutely.
So hold on one second, and I'm going to share some video.
Where was this? Was this from Little Cayman, or is
Nope.
This
This is last night here.
Okay, so this is an example of GoPro footage that you might get from the aggregation. One diver is operating it, and then you're seeing that's not going to taking the sleep there. And what we're doing when we go on these dives is we're sending a series of divers down. Each one of those divers has that same and they're trying to capture individual fish. With the GoPro, they're wide angle, which means you really want to get as close as possible to the fish so you can get bigger information. So the divers are swimming around the dive site, of course, always in buddy pairs, safety is usually important. But together, we're getting footage of these. Here's an example of this diver coming up on a fish, and you can see they're trying to get the sides to the pattern. So you often see, and here's a great example. You see how they're just off in the distance now. I hope you guys can see this. There's a huge band of Nassau up in the waterfall and are all milling around together. So that is that band that runs the length of the aggregation as far as you can see it in either direction and where this diver is. But there's also a bunch of fish that will up on the platform, and they're probably there because they're visiting a cleaning station. So lots of fish, they go to specific areas of the reef in order to get clean, like you would take your car into a cleaning station. Right. The fish will do that, so they'll come down onto the reef in small gobies and trips. And so it's there that we can get close to catch that big fish. But the vast majority of the fish that we're seeing right now are up in that band.
And is this where they're at all day long? Do you guys have to look for them, or do they move around? What, do they go down? Do they pitch their...
Yeah, it changes all the time.
And do you guys think that's related to conditions, or?
I think it's related to, first of all, you're asking great questions. And with science, we're still trying to figure out a lot of these terms, right? So we're already at the cutting edge of what we know for the behavior of the population. And yeah, so it does change from day to day. I think that when it's calm, like it is right now in this dive,
Yes.
they tend to band up because they don't have to fight against each other. Why do they band up? Well, they get together and one band above the ground, and they're checking each other out. And so it's like going to a bar, or they're visiting each other,
Right.
trying to figure out who they want to be there.
And which is really unique because they're a solitary fish the whole year, right? And so just on these few days of the year, they get together. But other than that, if they saw each other, they would fight.
They would like
We're territorial. They would protect their territory. Right?
exactly right, and that's part of why we think they take it very seriously. If you've seen an ass on the whole reef, this is not how they look. They have a very different pattern. You can see now tigers getting really close to the fish in the band. That's Dr. Christy Patton So they'll take on this different color pattern, and part of why we think they do that is to indicate to everybody else, like, hey, I'm a lover of fighting, right? I don't want to fight, right? I'm interested in reproduction. And so it's just a quick and easy way for them to signal that they're intentional. And you can see now as the diver's up in the band, this time of year as they move on that they get so focused on that reproduction, they ignore the divers. They're crazy close to these fish, and they just don't. You can tell.
So that was amazing footage, and thank you for that explanation. It looks like we have a new class that joined us, the Year 12 Marine Sciences class, that are headed to CCMI tomorrow. That's exciting.
That is awesome.
But as we're talking, if you guys have questions, feel free to raise your hands and or to put those questions in the chat. So the video that we just showed was last night, and I was out there on it. It was incredible, and I'm going to share more of that video on our blog later today. But you went out already this morning, didn't you?
We did.
And was it the same?
The same. The conditions are still amazing. You guys, it's hard to explain right now. Normally this time of year at the site that they're at, it was like pick a place on the map where you would least want to go diving because the conditions are terrible.
What makes it terrible?
It's deep.
Yeah.
It's right at the very edge of the island on a shelf break. It's where the currents get together and mesh, the waves stand up. It's just a very rough site, unpredictable. The current can pick up in a heartbeat. So it's a good place for reproducing if you're a fish, but it's a bad place to dive a lot of times.
Right.
But the last couple of days, the conditions have been absolutely incredible. No current whatsoever. And because of that, the fish are banded up. They don't have to fight the current. All they need to do is think about, hey, who am I going to choose? Who am I going to reproduce with? And so they're up in that band milling around, which makes our job easy because we can get into that band with our cameras and get all the footage that we need to in a short amount of time.
Now, is it just Nassau that are out there, or are other fish spawning there? Is it important
Right. Yeah. So I should say, and I know you all know this, but this is unique. There is nothing, what you just saw right now, there is nothing else in the Caribbean that's like that.
Right.
The Caymans are unique in what they've done to recover this resource. So they have these large spawning aggregations. So you all should feel proud of that because that's an amazing achievement and something that is nowhere else in the Caribbean. It's just here.
Yeah.
It is the case, though, that it's not just Nassau grouper that are out there. So a lot of the protections that the Caymanian government has put into place have also benefited lots of other species, including several other large grouper. It turns out that this nursery ward, as Dr. Croy likes to call it, this place of reproduction.
So yeah, it's like a nursery, right?
It's like a nursery. And so lots of different species come to this one site in order to reproduce. And species like yellowfin grouper, often called rock grouper here, black grouper, tiger grouper, we've even seen Goliath grouper out there, although we don't think they're there to reproduce. We think they're
You've seen Goliath grouper out there.
Goliath grouper are like the size of a car. They're huge.
They live up to their name.
Yeah. So lots of different species reproduce, not exactly at the same time, at the same night, but within a week of each other. They're all releasing their eggs at the same time. So it is a very critically important location for reproduction of lots of different reef fish species in the islands.
Now, last night I was out on the aggregation diving with you guys, a really incredible, amazing dive. And I was looking for sharks and I didn't see any. But when we came back onto the boat, Dr. Giddings was there and he had seen sharks. And I'm wondering, is that something that's common that you see out there?
All the time. In fact,
Really?
I just got off the boat, right?
Yeah.
And just came in here. Yeah, the last dive I was at, there was a reef shark, got an 8 or 9 foot reef shark out in the fish, just milling around. I have footage of it. Maybe we'll show you all the footage of it. And
We'll share that footage maybe tomorrow. We'll put it on the blog.
Yeah. So we regularly see sharks out there. I can tell you for a fact there are way more sharks out there than we can see.
Yeah.
Because sharks are really good at not being seen. They don't typically like to be around people.
Sure.
You see these movies about like the sharks coming and, that's just not how it is. Sharks don't want to be around people and they'll make themselves scarce when divers are in the water. But when it comes to aggregations, there's a tension. They don't want to be around people, but if people are in the fish and the fish are their food, you're going to see sharks. And so we see them pretty often.
Well, and we have a question from Cayman Prep, that just essentially what we were just talking about. How do the groupers deal with various predators in that area, like sharks? What happens?
Yeah, so a lot of them get eaten.
They do.
Or get bit. Yeah, we see it all the time. Especially on nights of spawning when they're actually releasing their eggs. And why is because when they're releasing their eggs, we think one female, there could be more. We're not sure yet. We're still doing science, right? We're learning all the time learning. We see one female go up and all males go up at the same, not all, but many males go up at the same time. And then they're all releasing their gametes. And that when there are many spawning bursts like that, the visibility drops because there's just so much stuff in the water. And between the visibility dropping and all of these grouper rushing up in this big mass, that's a great opportunity for a shark because the grouper aren't thinking about sharks. They're thinking about...
It's a buffet.
Yeah. So the sharks will attack right as the eggs are being released, which makes it's incredible to see, right? So you see all these groupers shooting up and here comes a shark, boom, and it attacks the big spawning burst. And we've seen that happen, and a shark will come out with a big grouper in its mouth, and it's jumping on the grouper, and its eggs are spewing out. It's crazy to see it. It's true.
We actually saw there. I can't remember. It was a couple of years ago. We were coming back from one of the dives, and one of the boats had found a grouper that had been chomped on. Like they saved him. Right.
That's right.
And that was essentially what had happened with that grouper. It looked like it was a shark bite.
He'd been bit. We see fish with wounds all the time.
I see.
I don't think. I mean, is it going to be a huge population impact? Probably not. I mean, it's nature. Sharks got to eat, right?
Well, and I've seen plenty of video for doing this project where you see the sharks, dive into the ball but miss. Like they're not always catching the fish.
They miss more than they catch.
Right.
Yeah.
Okay.
That's mostly what we see is the sharks not being successful. Poor sharks.
Cayman Prep wants to know if you've ever been bitten by a shark.
I have not.
No.
Like we talked about, sharks don't want to be around humans.
No.
So even down there, even at the aggregation site, they have
Nassau
no
probably
interest.
looks a lot tastier.
Oh yeah. If you're a shark, you definitely. A Nassau looks tasty. A human, they know enough to know that humans are not the thing they need to be around.
Right.
Yeah.
Thank you so much for being here and sharing with us. We have one more guest that I want to bring on. Bryce, thank you.
Thank you all. It's great to see you.
So there are other researchers here on the project that are doing various things. And like I had said before, we don't just see Nassau grouper out on the aggregation. We see other types of grouper and other fish. So I want to invite Janelle to come up and talk about, I believe tiger grouper is what you're working on. Is that, yeah?
Yeah. Well,
or you can talk
Yeah,
about
it's
whatever
fine.
you want that you're working on the project. So could you introduce yourself and say, how you got here, sort of the pathway that you got to here first?
Yeah, so I am a full-time graduate student at Oregon State University.
Woo!
I'm in Scott Hippel's lab, and he's another, PI on the project. And within under Grouper Moon, there are many different research projects happening at the same time. We already talked about fish faces and stereo videos.
Yeah, so it's not just, they're not just
Right.
out there counting. Like there's much
there's
data
many different
that's,
things happening. Yeah. And so my avenue really looks at the early life history stages of Nassau and tiger grouper, and yellowfin grouper as well. And early life history stages, meaning like literally once they are spawning and they become fertilized eggs, that's when I'm really looking at them and seeing how they progress over the next like week or so. And basically I'm seeing how climate change can impact, or more specifically increasing temperatures can impact the fish at those early stages.
so how do you measure that? Like what is it that you actually do to study that?
So I look at a couple different things. Some is just like straight morphometrics.
What does that mean, morphometrics?
So once we have the larvae, we store them in different preservatives, take them back to a lab, and we can use a microscope to take pictures of them.
So you've created a lab actually,
actually
in the room that we've rented here on the island. And you guys, I'm going to take pictures of it and I'll post it on the blog so you can see. But what you're actually saying is when they spawn, you're going to go out and capture some of the eggs.
Yes.
And then what will you do with them?
Yeah,
in the tanks?
We'll bring them back to the lab, put them in different temperature treatments in each aquarium tank that we have built in this Airbnb kind of.
Yeah, I mean, it's a makeshift research lab, right?
Yeah, exactly.
It's pretty awesome.
Yep, and so each day we'll pull out samples and store them in these preservatives so we can take them back to another lab back home and see. We can take pictures of them and see how is like their length changing, how is their like, are they pigmenting earlier? Or
what does that mean?
Pigmenting is when you start to see coloration in their body.
So that's kind of like development.
Yeah, development.
Stage.
You're right. We
Okay.
can see how they're developing over time, over usually like a 6-day time window.
So this isn't your first time here. You were here last year,
yes,
right?
Yeah.
So tell me a little bit about did you have any ahas or what did you take away from last year and what are you hoping to build on this year?
Yeah, so last year, I did notice there are a lot of developmental changes in different temperatures when they're.
So you did see that when you had them in different temperatures, you were seeing that they developed differently.
Right, but another interesting thing is that we target individual females when collecting. So we have samples from 4 different females, and we can see that some individuals produced from certain females do better than others. And so that leads into like my second question is like, which involves a lot of genetics, but we're basically seeing like, do certain females have better genes essentially than others? And if so, it's really important to keep the population as big as possible.
Right.
So the future populations have those good genes that can deal with climate change issues a lot better. And so it's a really big project to support the fight for keeping the population so big so they can do better with other issues. Like a lot of the work before has been done to get the aggregation site as big as possible so it can recover. And now that we're seeing recovery, especially here on Little Cayman, we're looking at other questions like climate change and other issues that could be happening out here.
Awesome. So I know that we have some year 12 students on here that might be thinking about going into marine sciences and so I was wondering if you could just tell a little bit about your story, like what inspired you, what were the first steps that got you to be here?
Yeah, so I grew up in more of a rural area, nowhere near the ocean. Just a couple.
Where were you?
Pennsylvania, rural Pennsylvania.
Okay.
And so I didn't even know this was an actual career possibility. I had no idea people did this for a living. I didn't know you could make money off of it.
So what happened?
I ended up going to school out of state, and I went to school in Virginia, and it's really close to Virginia Beach and right on the water.
Okay.
And I was there actually to study biology on a pre-med track because I like science. I just didn't know what to do with it at the time, and everyone said be a doctor. So that's what
everyone said. Be a doctor. Okay.
Yeah. So that's what I went for. And my first year, I hated it. I
Really?
didn't like anything that I was told to take, sort of. But I took a few electives. We had a department called Marine and Environmental Science. And so I took a few electives in that class or in that department. and pretty much switched my major as soon as I
Really?
realized how great it was. Yeah. And so I was volunteering in a lab over there, which brought me to a conference called American Fisheries Society. It's an international conference on a bunch of different scientists that study fish.
Mm-hmm.
And that is where I met Scott Happel, who works on this project.
Mm-hmm.
And we talked a lot about my personal interest for research and his, like what he does in his lab. And I also love to travel. So as soon as I heard like what he was doing, I was like, I have funding, I can come. Like,
Yeah.
yeah, like please take me on. And it worked out ever since then. So I moved to Oregon to join that lab, and I didn't get to come to Cayman my first year, mainly because I started during COVID. So 2020 really.
In fact, one of the other students wasn't able to come because they got tested for COVID.
Mm-hmm. So that is one big challenge. Nothing goes as planned half the time. You have to be very adjustable in this field, I feel like. But
you got to roll with it.
Yeah, I kind of roll with the punches. And so, but it did work out in the end. I didn't get to go my first year, but last year I got to go. And I was here in January and February because it was a split moon.
Mm-hmm.
And then I also got to come back this year to kind of expand my studies a bit more. Last year, I primarily focused just on Nassau. This year, I'm also looking at potentially tiger grouper as well.
And that's something we do see a lot of tiger grouper out there on the aggregation. And I see a couple of questions. One of them, what would happen if you caught a Nassau grouper? Well, my hope is that you wouldn't be out there catching Nassau grouper right now because it's during the spawning season. So people are not supposed to catch them. And if you happen to, I would hope that you would release it immediately. But outside of the spawning season, you can catch grouper. And there's catch limits that have been created by the DOE and Reef as well for, you can't catch grouper that are too small because they need to at least be old enough to be reproductive age before you can catch them, those types of things. So you can catch them, but not right now, not during the spawning season. And I would tell you the exact dates of like when the catch season ends, but all of our DOE folks just took off. So I will find that out and I'll post it on the blog for you. What are the cons of working in the marine biology industry? I'm going to let you talk about that. What do you think?
Well, I guess I love it. So,
so you're biased.
Yeah, I guess I'm biased, but it is a long journey, especially if you're interested in the research. Most or a lot of other jobs, you graduate college and you kind of just go straight into your full-time job. And here it's a bit different. If you're interested in the research, you end up going to grad school and potentially another time in grad school and maybe a postdoc. It could be.
So how long, what, is there an average number of years of school or does it really vary depending on what you're doing?
I think it varies. And that's,
Okay.
I probably should have explained that too. Every person does not go through the same situation. What I did is probably completely different than what Bryce just did when he was talking,
Sure.
or what Coy did when he was talking. So we all go through very different paths. But a lot of the time, people do end up going to grad school, and that can take a bit of time depending on what degree you're going for. Like a master's could be 2 to 3 years, or a PhD could be 4 to 6 years.
I mean, so I would think a con then might just be it's expensive.
Yeah.
Right. You're many years in graduate school. Right.
Yes. And it's not as, I guess, laid out for other professional occupations, like medical doctors,
The
it's
track is
specific,
pretty clear.
Yeah. You're going to undergrad, you go to med school, you do a residency, etc. Whereas something like this, you may do a master's, you may not. You may go straight to a PhD. You could do a postdoc, maybe you don't. There's many different paths that you could take, and trying to navigate that and figure out what's best for you can sometimes be difficult because everyone does something different, and there's no right or wrong way to do it. It's just kind of what works for your situation.
Wow. If you breed 2 groupers with good genes, could you make a super grouper? I love that question. I don't know. What do you think?
Well, that's the, I guess, whole idea of my project. We're trying to see if certain individuals can express certain genes in a better way than others. And if we keep those genes in the population, that, it gives them a better fighting chance. So is it a super grouper? I don't know if we would call it that, but it does,
But they're healthier.
but it does,
grouper,
yeah,
right?
it does give those individuals a better chance at fighting issues like climate change and increasing temperature.
Well, and that's why we want to have such a big aggregation, so that there is a wide variety of genetic material that's being shared. So you do have lots of healthy grouper.
Yeah.
So what's next for you? Well, you'll do this here, and then what will you do? What will you do with the information, the data that's collected here?
Right. So I will bring everything back to my lab at Oregon State University. And that's when I go through, take pictures of all the larvae we collected. I'll do a lot of the gene expression work I'm doing and bring them to like this huge genetics lab, extract RNA. I'm sure that's too detailed.
Yeah. No, it's good. We've got year 12 here.
Okay. Yeah. So we'll do RNA extractions from each individual larvae, and then we can run it through this PCR, which basically amplifies the RNA, and we can see, how much of that protein is being expressed in certain individuals in temperatures compared to others. And so we're really just comparing to see if we, how much the heat shock proteins are being expressed in higher temperatures compared to normal temperatures, and then if it's different between each female as well.
I see.
Yeah.
So will that turn into a paper for you, or does that end up that data end up somewhere?
Yes. So, and when you become a scientist, publishing is super important. So you're going to I will eventually write this up, and it'll hopefully be published and go somewhere for future scientists to refer to and to do their, they can reference this paper for their studies and other projects as well. And it would be cool to also do similar work for other species, like we said before, like tiger grouper or yellowfin as well.
Awesome. Thank you so much for being here and sharing with the kids, you guys. Thank you also for your questions. Oh, I've got a question. Can grouper get cancer? I don't know. I mean, I think that fish do get cancers. I don't know the exact answer to that question, but I thank you for it. And I'm going to do a little research on that afterwards, and I'll post a more comprehensive answer on the blog. But thank you for that question. Thank you so much for being here. That's going to be it for us today. Please join us tomorrow, same time, same place, 11 o'clock. We have a whole nother slew of scientists that are going to be here that you're going to get to talk to. We're going to look at some of the really great scientific tools that are being used out here on the aggregation to study the Nassau grouper. We'll have some more footage, and then be sure to join us on Wednesday, where we will be going live from Bloody Bay Wall, a famous dive site here on Little Cayman, and you guys will get to see some of the beautiful and amazing marine life that we get to experience out here. So thanks you all so much for being here. Cayman Prep, thank you for hanging out till the end. Have an awesome day. I'll see you all tomorrow. Go Grouper Moon.